PCR. Okay, here's what you can write down. This is what it stands for. PCR stands for Polymerase Chain Reaction. Polymerase Chain Reaction. I know it's only 8:17 on a Wednesday morning, and you've slept since then, but who can remind us what polymerase is? Anybody remember what polymerase is? Excellent. It's an enzyme. It ends in ACE. That's a hint that it's an enzyme, right? It's an enzyme. Does anybody remember what that enzyme does? Angie? Yeah. It replicates DNA. It replicates DNA. Remember, okay, here's our DNA, okay? Double-stranded DNA, and it unzips. Remember that? And then new nucleotides pair up alongside this side of the DNA. And new nucleotides pair up along this side of the DNA. Polymerase is that enzyme that does that. That grabs the free, lonely, unattached nucleotides and pairs them up. It's kind of like a matchmaker, remember? DNA polymerase, that's the enzyme that does that. Now, so we know what polymerase is. Chain reaction. Okay, what do you think of when you hear the term chain reaction? Chain reaction. What do you think of?
Like one thing happens, and then it leads to another thing.
Excellent. One thing happens, leads to another thing, leads to another thing. When you were a kid, did you ever set up dominoes in like rows, elaborate formations, and then you knock one over and they all go down like a chain reaction? One thing leads to another, leads to another. Chain reaction. And in a lot of chain reactions, as it gets going, it really speeds up. Well, this is called polymerase chain reaction because basically what it is, it's a technique that you guys are going to do in the lab with your DNA. And what you're going to do in a test tube is you're going to replicate your DNA and you're going to make millions of copies of it. It's basically DNA replication gone crazy in a test tube. And it's like it takes off like a chain reaction. Now, let's think about this. If you have one DNA molecule, and that one DNA molecule, okay, here it is, one DNA molecule, it splits apart, it replicates, then you got two DNA molecules, right? Then when they do that again, you have how many? 4. And then when they replicate, you have how many? 8. I know this is challenging early in the morning when you haven't had a lot of coffee. Those 8 replicate, then you have how many? Those 16 replicate, then you have how many? 32, and then how many? 64, then 128, and then 256, and then 512, and then 1,024, and then 2,048, and so on. It takes off exponentially. Chain reaction. Okay, in a few hours you've got billions of copies of that particular DNA. So that's really what polymerase chain reaction is. DNA replication gone crazy in a test tube. If you have just a tiny little smidgen amount of DNA, you can use this technique in the lab to make billions of copies of it, so you have enough that then you can run it through a gel and do a DNA fingerprint. Okay? Now, I found some interesting ways that PCR has been used. When the investigators started out with just a teeny tiny smidgen amount, interesting ways, and they want to make billions of copies of it so they have enough to run it through a gel and do a DNA fingerprint. These are some ways that this has been used. Okay. You ever heard of Jesse James? Jesse James was an outlaw in the Old West. Like bank robber, stagecoach robber, Jesse James. And of course he's dead, obviously. This is in the days of the Old West. A notorious outlaw. And he was buried in a grave, of course, and over the last close to a couple hundred years, people have thought, that's Jesse James' grave, and other people thought, no, that's just a myth, that's just a legend, that's not really his grave. So guess what? They dug up his remains. And now, if somebody's been dead since the 1800s, there's not much DNA left, right? In their remains, in their coffin, there's not much left. Just trace amounts, just a smidgen is left, that's all. So they isolated just that little smidgen of DNA from his remains and then did PCR on it to make billions of copies of it, so they had enough to make a DNA fingerprint. And then they compared it to his living known relatives, and sure enough, it was Jesse James. So that's one way it's been used. And here's another. It's been used on roadkills and carcasses washed ashore to identify locally threatened species. If you've got a carcass or a roadkill, been dead for a while, not much DNA left in there. So they've done PCR on it to make billions of copies of it, so they have enough to do a DNA fingerprint. Oh, listen to this one. PCR has been used to amplify DNA. Now, amplify DNA means make billions of copies of it using PCR. Okay, it was used to amplify the DNA from a cremated man. Okay, so he's been dead and cremated already. He's gone. But there were skin cells left in his electric shaver. There were skin cells left in his electric shaver, and they isolated the DNA from those skin cells and used it then to diagnose an inherited disease in his children so that they could compare his DNA to his children's DNA. The brain of a 7,000-year-old human mummy in Egypt. A 7,000-year-old mummy in Egypt doesn't have much DNA left in it, just a smidgen. The digestive tracts of carnivores to reveal food web interactions. Interesting. Products illegally made from endangered species. Okay, that's interesting. What are some examples? Can you think of any endangered species, like from Africa, maybe? Rhinos. Okay, so let's say somebody's trying to sell, leather bags or purses or something, and somebody suspects that those were made from the skins of rhinos that were poached by bad guys, poachers. There's not going to be much DNA in the leather in that bag or purse, just a smidgen. But with PCR, they're able to take that smidgen and make billions of copies of it so that they can make a positive identification and say, yep, that really came from that endangered species. Evidence to try to put poachers away, for example. Now, the last one here that I thought was kind of interesting, they used PCR to make billions of copies of DNA that was isolated from the guts of genital crab lice. On a rape victim. Does that make sense? So in other words, these lice jumped from the rapist onto the victim. The investigators isolated these lice from the victim. They took the lice back to the lab. They had a suspect. they extracted from the guts of these lice cheek cells, not cheek cells, skin cells that the lice fed on. See, when lice get on you, they start to chew on your skin cells, and that's what makes you itch. Okay? So in other words, they isolated skin cells from inside the guts of these lice that were on the rapist. So the lice had the rapist skin cells in their guts. They isolated the DNA from those skin cells in the guts of the lice, did PCR to make billions of copies of it, did DNA fingerprinting, and it matched the DNA of the suspect. So they were able to put him away because of that. That's kind of strange. Clever detectives. So anyway, those are some ways that that's been used. PCR. Now, I want to show you how it works, because you guys are going to be doing this in lab next week. That guy right there, anybody know who that is? You get bonus points if you know who that is. Probably don't know who that is. Okay. His name is Dr. Cary Mullis, born in 1944, still alive. Still alive. He invented PCR. In fact, he then got a Nobel Prize for it in 1993. He invented PCR. Now, I'm told that he was really into surfing. Lived on the West Coast. He's a surfer. Really into surfing. I mean, he's a scientist, a researcher, but in his spare time for fun, he goes surfing. And the story goes that he was driving home on the California coast one moonlit night, and the idea just like popped into his head. Got this really good idea about how to make billions of copies of DNA in a test tube. And that's kind of the beginnings of how he invented this technique called PCR. PCR, DNA replication going crazy in a test tube. Okay, I already told you about that, right? Let's look at how it works. Now, to do this, to make billions of copies of your DNA in a test tube, you have to put it in a special machine like this. Now this machine's called a thermocycler. It's a thermocycler. It's got a computer in it. It's got like 24 little holes in here to put tiny little tubes in. The test tubes are plastic and they're really small. For 24 tubes. So 24 student samples can go in here. And program this thing. And what it does is it heats up the sample to almost boiling, and then cools it down, and then heats it up again, and then cools it down, and then heats it up again, and cools it down. That's why it's called a thermocycler. You program it to do that. Okay? And the reason you heat up the DNA to almost boiling in your little tube in the thermocycler is to make the DNA unzip, like that. See, if you heat up DNA, it'll unzip. Now, interestingly enough, it doesn't destroy the DNA, but it will make it unzip. Now, here's the problem, though. If you want to replicate and make billions of copies of your DNA in the little tube in there, the polymerase enzyme that replicates your DNA will be destroyed when you boil it, when you get it up close to boiling. So we know how in the thermocycler to heat it up to almost boiling so your DNA unzips, but then the problem is the polymerase enzyme that's supposed to replicate the DNA is destroyed at those high temperatures. So Cary Mullis got the idea, how about if we get polymerase enzymes from these really weird primitive bacteria that live in hot springs. So we use a polymerase not from humans, but we're going to use a polymerase from these bacteria when you guys do the lab. you'll use bacteria polymerase that can survive high temperatures, and it will know how to replicate your DNA just like your own polymerase would know how to replicate your DNA. Okay? So write this down. The bacteria that we got the polymerase from is called Thermus aquaticus. That's the scientific name of the bacteria. Thermus, bless you, aquaticus. Thermus aquaticus. Thermus means what? Thermometer means heat. Aquaticus means they live in the water. Okay? The polymerase that you get from these bacteria is called Taq polymerase. TAQ, that's just for Thermus aquaticus, TAQ. Taq polymerase. That's the kind of polymerase enzyme we're going to use. And has any of you guys ever been to Yellowstone National Park? Anybody ever been there? It's beautiful country, it's out in Wyoming, but it's very close. The volcanic activity is very close to the surface of the earth out there. And so there are these hot springs. Like, see that pool right there? See the steam coming off of it? You wouldn't want to fall into that pool because the water's almost boiling. It's almost boiling. Now, they have boardwalks around here with signs that say, "Danger, danger, stay on the boardwalk." Because supposedly some guy several years ago wandered off and started walking out here and fell through, and it cooked him. It's hot water. But the reason it's got that kind of turquoise color is that's caused by bacteria that live in there. There are bacteria that live in that water that's almost boiling. They're called Thermus aquaticus. They're very primitive kinds of bacteria. Very old, very primitive, strange bacteria that can live in extreme environments. And so Kary Mullis said, let's just get some bacteria out of there, isolate the polymerase enzymes from those bacteria. It must be able to withstand those high temperatures because, my gosh, they live in there, these bacteria. So that's the kind of enzyme we're going to use. Thermus aquaticus. All right. Let's think about this for a minute. Daquan, I bet you could think of an answer. If you were going to put your DNA in a test tube and make billions of copies of it, besides your DNA, what ingredients would you need to put in there? We can figure this out. What's the recipe going to be? Okay, we're going to have our little tube, we're going to have our DNA in it. And we want to make billions of copies of it, so what should we add to the tube? I already just mentioned one of them.
DNA.
Oh, your DNA. Good. Excellent. We're going to call that the template.
The Taq polymerase.
Excellent. Taq polymerase. Yeah. Taq polymerase. There it is. We're going to need that. And we're going to need, how about this? Lots of free nucleotides. Doesn't it make sense we want to put a whole bunch of A's and T's and C's and G's in that tube? Building materials to make more DNA because we're going to make billions of copies of it. And, something we're going to be talking about here in a little bit called primers. Little molecules to get the reaction started. They're called primers. Those are the ingredients. Oh, and of course your DNA, your template. Okay, let me show you how this works. PCR, step by step. There are 3 words you need to write down. These are the 3 steps. Denaturing, annealing, extending. Okay. Denaturing just means splitting apart the DNA. Okay, watch. Let me show you what denaturing looks like. You ready? Here's your DNA. That's denaturing, unzipping the DNA, splitting it apart, okay? That's the first step, denaturing. We do that in the thermocycler. It heats up to almost boiling, and that heat causes the DNA to denature or unzip. The second step is called annealing. If something anneals to something else, it's basically sticking to it. So the second step's called annealing. That's where, after the DNA denatures, the thermocycler is going to cool down just a little bit to allow these molecules called primers to attach to the DNA. And it will also allow the polymerase enzymes to latch on. And then the thermocycler will heat up just a little bit, not quite as hot as the first temperature. Heat up just a little bit to the temperature that the polymerase enzyme really likes. And then the polymerase is going to replicate the DNA. It's going to replicate it, extend the new strands. So that step's called extending, when the DNA is actually being replicated by the polymerase. So those are the three steps. I'm going to show you what they look like. All right, this is what they look like. Now, I need to show you something here. Before we get into detail into those steps, I'm going to draw up here your DNA that you're going to extract. Okay, let's use. Okay, let's say we extract your DNA. Lily?
How long does this process take?
The whole process?
Just in there.
It's going to take about 3 hours. So when you guys next week put your samples in here with all those ingredients, we'll let it run for about 3 hours. So after that time period, you guys will be in some other class, and I'll just take them out. You will have labeled them with your name, and I'll store them in the freezer until the next time we can continue the lab. It takes about 3 hours in there.
And then how many times does this switch back and forth?
Excellent. About 40.
40?
Yeah, 40 times. So it'll go, let's go back to those steps. It'll do denaturing, annealing, and extending. That's one cycle. And at the end of that one cycle, if you've got one DNA molecule at the end of that one cycle, then you'll have two. And then it'll go through those steps again, and that'll be the second cycle, and you'll have four.
Okay.
And then you go through it again, that'll be the third cycle, and then you'll have 8, and so on. By the end of 40 cycles, you can do the math if you want. You can do it in your head right now. You're going to have lots and lots of DNA. But here's how this will work. Now, this Friday, you're going to actually do the first step in this whole lab procedure, where you'll extract your DNA from your cheek cells. You get to spit into a cup. Okay? You'll do that this Friday. And then you'll do some other things to it, and I'll hand out the lab here in a little bit. But let me show you what your DNA is going to look like then. Here it is. Long thread of DNA. This is all the DNA from your cells, your cheek cells. Now I've got this memorized, so I know how to draw this. I can do it with my eyes closed. Okay, there it is. All right, that's your DNA. Pretend. Now, that's just a smidgen amount. Now, this is DNA from like millions of your cheek cells. That's what you're going to spit back into the cup. That sounds like a lot, but there's really not a lot of DNA there. I mean, if you took that DNA and you put it into a gel and did electrophoresis and even dyed it with a stain, you wouldn't see it in the gel, be invisible, because it's such a teeny tiny amount, even though it's from millions of your cheek cells that you spit back in the cup. So we have to use PCR to amplify it, make billions of copies of it, so there'll be enough that'll show up in the gel. Now, the only thing is, You're not going to do PCR on all of your DNA. We're only going to make billions of copies of one little piece of DNA. Let me see if I can find it. Oh, here it is. It starts here and it ends right there. Just that little piece of DNA right there. That's all we're interested in. We're going to do PCR on just that little piece right there. Now, we've got these special little single-stranded DNA molecules. They're really short. They're called primers. Primers. That means they're going to prime the reaction. They're going to get things started. And these primer molecules are going to latch on. Watch this. They're going to latch on right here and right there. And they're going to get the reaction started. And then this little piece that we're interested in will replicate. Then we'll have 2 copies. And then after another cycle, we'll have 4 copies, and then 8, and then 16, and then 32, 64, 128, 256, 512, 1,024, 2,048, and so on. See how it's replicating just that little piece? Pretty soon we're going to have billions of copies of that particular little piece that we are interested in.
What happens to the rest of the DNA?
It's still there. But see, then when we squirt all this DNA into a well and we run it, do electrophoresis on it, the only thing that's going to show up is this particular piece because there are billions of copies of it. All the rest of this is not even going to show up. Even though it's going to be in the gel, it's not even going to show up. It'll be invisible. Does that kind of make sense? So when we do PCR, we only do it on a particular little piece that we're interested in. Now, what little piece are we interested in? Can I show you? I'll show you. I'm going to show you what little piece we're interested in. What little piece we're going to amplify and make billions of copies of. Okay. Remember your chromosomes. On chromosome pair number 16, at the tip of chromosome pair number 16, you have a little sequence of DNA. It starts here and it ends there, and it's 641 base pairs long. Actually, you got 2 of those because you got 2 chromosome 16s. So there's the other one. 641 base pairs long. Everybody's got 2 of those. Now that's a particular little piece of non-coding DNA. Okay, it's not part of a gene, it's non-coding. A little piece of DNA. Maria, that's a little piece of DNA on the tip of your chromosome number 16. And there's the one on the other number 16. Now, some of us have a little piece of virus DNA inserted in that sequence. A little piece of virus DNA. Some of us have it and some of us don't. And I'll show you what it looks like if you have the virus DNA inserted in there. If you have the virus DNA, that stretches out that important sequence there. So that kind of looks like this. All right, I'm going to make the virus DNA red. Actually, it has a name. It's called an ALU insert. It's inserted right in that 641 base pair length of DNA. And that little piece of virus DNA is 300 base pairs long. So, if you've got that virus DNA inserted in this piece, then the piece ends up being how long? 941 base pairs long. Yeah. Okay. if you've got that virus DNA inserted into that piece of DNA on the tip of your chromosome 16, then that piece of DNA suddenly becomes 941 base pairs long. Now, if you've got that virus DNA inserted in that piece of DNA on your chromosome 16, it's no big deal. It doesn't matter. Doesn't hurt you, doesn't do any harm. It's just one of those little pieces of virus DNA that we carry around, some of us, and it uses us to reproduce it, to make copies of it. Because every time our cells multiply, they make copies of that little virus DNA. So it's a little like hitchhiker, just hanging around in us, using us to make copies of it. Okay? Now, some of us have that virus DNA in one of our chromosome 16s. Some of us have it in both of our 16s. Some of us have it in neither of our 16s. Some people just have that 641 base pair piece on one chromosome 16 and another 641 base pair piece on the other chromosome 16. And when we do the DNA fingerprinting, we're going to find out who's got the virus and who doesn't. Okay? Now, Angie, if you've got the virus, remember, it means nothing, no big deal. Having the virus versus not having the virus is sort of like brown eyes versus blue eyes. No big deal. Doesn't matter. Just kind of interesting. It gives us an excuse to do DNA fingerprinting using PCR. So, Angie, if you've got the virus on one of your chromosome 16s but not on the other one, then we're going to say you're heterozygous. Okay? If you've got the virus on both of your chromosome 16s, then we're going to say you're homozygous positive. You've got it. If you don't have the virus on either one of your two 16s, then we're going to say you're homozygous what? Negative. Okay? So, the primers, these little single-stranded pieces of DNA that are going to latch on to both ends of this target sequence, they're going to bracket that, you see, and target it, and that's where the Taq polymerase enzyme is going to replicate just that piece of DNA, just that piece. Now, can you see how if you've got the virus DNA, when you run the gel, this piece right here is not going to move as far in the gel as these shorter pieces, right? And that's going to be the basis of how we're going to be able to tell by looking at those bands in the gel whether you've got the virus or not. Is this stuff making sense? This is all new, I know. Claudia, is it making sense?
Yeah.
Sort of?
Yeah.
Okay. All right. So let me just show you how this actually works, kind of up close and personal. Show you how it works. Okay, here's the first step, denaturing. This is your double-stranded DNA. Now, let's say that's the target sequence right there. That's this piece right there. Okay? The machine, the thermocycler, is going to heat the tube up to, well, 94 degrees Celsius. That's almost boiling. What, in Celsius, what's boiling? 100. So 94, that's pretty hot. Okay? And that's going to cause the DNA to unzip. See? It unzips, it denatures. And then the machine will cool down to 60 degrees Celsius. That lower temperature allows the primers to attach. See, here's a primer right here. It's a little single-stranded piece of DNA. They come in the kit. Little single-stranded piece of DNA that will hook on, latch on to this end of the target sequence. And then there's another primer that will hook on to the other end. And so it brackets the target sequence. So the primer is going to bracket there, there, and there. it tells the polymerase where to start replicating. At 60 degrees, the polymerase molecules, here they are, Taq polymerase, they're going to attach to the primers here, and they're going to get ready now to replicate the DNA. The machine then will heat back up to 72. Are you getting this? It'll heat up to 72. 72 degrees is what the Taq polymerase enzyme really likes to work at. And so that Taq polymerase is going to replicate that DNA. That's called extension. It's going to extend it. And the polymerase on the other side is going to replicate that DNA. So here's our extension. Now you see we've got 2 DNA molecules where we just had 1 before. And then, like we talked about, Lily, just a second ago, or a couple minutes ago, the machine's going to repeat that cycle 40 times inside there. It'll take about 3 hours. And that's how it works. That's how polymerase chain reaction works. Let's
A sample of chromosomal DNA, also called genomic DNA, can be used as the starting material for the polymerase chain reaction, or PCR. With PCR, an investigator can amplify a single copy of a DNA segment in billions of identical copies. The DNA segment of interest, or target DNA, is indicated in red. In addition to the target DNA, a PCR reaction contains several other ingredients. These include free nucleotides, DNA primers, and the enzyme Taq polymerase. The primers are typically about 20 nucleotides long and are complementary in sequence to the ends of the target DNA. Taq polymerase is derived from hot springs bacteria and can tolerate the intense heat of a PCR reaction. A PCR reaction lasts several hours and typically consists of 20 to 35 repeating cycles. A cycle begins by heating the reaction mixture to 95 degrees Celsius. The heat denatures the DNA, breaking the hydrogen bonds that hold the strands together. After denaturing the DNA, the temperature is reduced to around 60 degrees so that the primers can form hydrogen bonds, or anneal, with their complementary sequences in the target DNA. Note that the primers and the target DNA follow base pairing rules. An adenine, A, pairs with a thymine, T, and a cytosine, C, pairs with a guanine, G. In the next phase, the temperature is raised to 72 degrees Celsius. Taq polymerase functions optimally at this temperature and begins polymerization, adding nucleotides to the 3' end of each primer attached to the DNA strand. After one complete cycle, there are 2 double-stranded copies of the target DNA. The PCR reaction mixture contains many copies of the primers and an abundant supply of nucleotides to perform many additional cycles. After a second cycle, there are 4 copies of the target DNA. After cycle 3 is finished, there are 8 copies of the double-stranded target DNA sequence. Note that only 2 of the double-stranded copies consist of just the target fragment. The others also include spiking DNA regions. As the number of cycles increases, the products consist of a greater proportion of fragments with just the target DNA. After 4 cycles, half of the fragments consist of just target DNA, and half of the fragments also contain flanking DNA. With each additional cycle, the number of copies of our target sequence doubles. At the end of cycle 25, there are more than 33 million copies of this double-stranded target region.
Okay, that makes sense? Sort of? See why it's called a chain reaction? Because the number of copies starts to increase exponentially really fast. Here's another look at it.
Polymerase chain reaction, or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the primed sequences and adds nucleotides to extend the second strand. This completes the first cycle. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
Okay, that's just another look at it. That's basically it. That's all the new information. Okay, I know it's a lot. Okay, and you're gonna have another chance to look at it again. But there are the steps again. Those 3 steps: denaturing, annealing, extending. Got that? Now I want to show you a video clip. It's kind of cool. It's actually a commercial. It's a music video. And it's a commercial for a company that sells science equipment. It's advertising, it's an advertisement for a new thermocycler. And they got all these scientists, all these geneticists, actual PhD scientists, geneticists, to do this video, to make this video. And they gathered together a bunch of scientists who could sing. And they produced this music video. It's actually an advertisement for a new thermocycler. Check this out.
There was a time when the amplified DNA you had to grow tons and tons of tiny cells. Oh, and along came again and Dr. Carey Morris said you can amplify in vitro just as well. Just brings you down with a pulver and some primer. Nucleotides and polymerases too. Denaturing, annealing, and extending. Well, it's amazing what heating and cooling and heating will do. PCR.
Out there doing PCR, BioRad salutes you with the all-new 1000 series thermal cycling platform.
I have got to get me one of those. Does that make you want to go out and buy one of those thermocyclers? No? All right. Let me give you guys a copy of the lab that we're going to be doing on Friday. So you'll have it. Now, this is just the first step. It's a lab that's going to take us like 4 class periods to do. So first we've got to extract your DNA. Then we have to use PCR to make billions of copies of it. And then we have to do electrophoresis, run your samples through a gel, stain the gels, and then look at the gels and try to find out what your genotype is. So it's going to take us several days, but this is the first step right here. And this just explains how you're going to extract your DNA. It's a little bit more involved than what we did before Christmas vacation. Remember when you extracted your DNA and put it in a little vial? It's a little more involved than that. You're going to get a little more of a pure sample, a little more technical. So what I really need you guys to do before you come to class on Friday is read through this carefully. Okay? Read through this carefully. And that is all. Lily?
Can you put the schedule up for today up on the board?
Oh, the schedule?
Or if I can take a picture of it or something.
Yeah, I'll just,
Take it.
yeah, here it is. And we can pass this around if anybody wants to take a look at the revised schedule for today.
Hey, Mr. Ruhl.
But that's it for today. I just wanted to talk to you a little bit about PCR before we start to extract the DNA on Friday.